in the plant space biology world for the first hour, so sort of the 8 to 9 o'clock hour. And then from 9 to 10, we'll hear about animal space biology work. And so I've invited some outstanding speakers, some real leaders in the field who have had some really interesting data from their space flights to come speak to us this morning. So just to remind everybody, we're going to, in this session, handle questions in the same way we did yesterday, using texting. And so if we have that number, we could maybe project it. If not, I will tell you what it is. 855-855-1413 is the phone number. If you have any questions during the session, please text them to that number, and I will be monitoring the text log. All right, so we might as well go ahead and get started. So we're going to first start, as I mentioned, with the plant biology portion of this symposia. And so I'm actually going to turn the mic over to Dr. Howard Levine. You all know him. He's a past president of ASGSR. He also is at the ISS National Lab, working in the research office at the Kennedy Space Center. And so Dr. Levine has been involved in space biology on the plant side for many years. He's actually been involved in over 90 space flights as either the PI or as part of the space team. So he really is a really authority in this field, in this area. And so I've invited him to introduce this portion of the symposia and also our speakers for the plant biology portion. So with that, I'll turn it over to Dr. Levine.
Thank you, Josephine. Okay, so I'm going to apologize right away. I'm going to rush through these slides so that we have more time for our distinguished speakers. But what I decided to do was to give you a very brief overview of the advanced plant habitat hardware so that you're familiar with it prior to their talks. Okay, so essentially this is it's a quad locker unit that's up on the International Space Station. We have another flight unit at Kennedy Space Center that we do our ground control experiments within. And you can see in the lower right, it has a very large chamber that's some wheat growing within it. And it's really about the same size, the growth volume, as the veggie unit. We have 2 veggie units on ISS. But the difference here is that it's under controlled conditions. So it controls temperature, relative humidity, CO2 concentration. It scrubs volatile organic compounds, especially ethylene. So for the experiments that require a controlled environment, this is our flagship piece of hardware. It has a variety of LEDs so that you can manipulate the quality of light and the quantity of light. It has the capability of going up to 1,000 micromoles, although very few experiments would ever go above, let's say, 600 micromoles. So the heart of it is, at least for the science folks, the science carrier. So in the upper left, you see an empty science carrier. Upper right, you see a science carrier with Arabidopsis seedlings that have developed, and it's very well outfitted with cameras. So bottom left, you see a top-down view of the plants. Bottom center is a side view, and bottom right is a side view with infrared camera so that you can see what's happening during the dark interval. So we did a validation experiment prior to our peer-reviewed research that you're going to hear about today. We grew Arabidopsis in the front half of it and wheat in the back. It's divided up, the science carrier, into 4 quadrants. Each of them is independently controllable, so if you want to have different wetness level set points or different substrate or whatever it is, you have that capability. But so I have a little video for you here. So you can see the Arabidopsis in the front and the wheat in the back. This is the hand of God harvest of the Arabidopsis, and then we let the wheat grow for a little bit longer. And then here's a little video for the second talk you're going to hear about, Carl Hasenstein. These are ground studies with his radish within the, on the left side, it's what we call the Delta EVT within the flight-rated hardware for ground control experiments. On the right side is a engineering design unit, so we also use that for ground studies, but just kind of a side-by-side comparison of radish growth from using the top-down camera over the, let's say, 28-day growth interval. And here are the radish, some of the radish that were grown during a particular ground study. And I think, you know, most of you have probably heard about the current experiment that's going on in APH with pepper plants on the ISS, and that's going very well. And I think yesterday they did a harvest, so we're very happy with that. And I know you can't read this, but you know, if you download the presentation, these are the specs for the types of environmental conditions that it controls. And many thanks. There were a lot of folks involved in developing the APH over time, the ISS Program Office, the Biological and Physical Sciences Division at NASA. So we're very thankful to them. And we've provided a reference on the bottom from that first validation experiment, Grow Out. So it kind of describes how that experiment went. It also talks about the capability of doing CO2 drawdowns for photosynthetic measurements. Okay, so thank you for that. So let's see here. So our first speaker for today is Dr. Norman Lewis. It gives me great pleasure to introduce him. He's from the Institute of Biological Chemistry at Washington State University at Pullman. And I've known Norman since the early 90s when Thora Holstead, you know, one of the major creators of this whole society, decided to use the now retired Plant Growth Unit spaceflight opportunity with 3 different PI groups. She divided it up. This was the Chromex-4 experiment, the 4th flight of the PGU, actually the 5th, but the 4th since it became a Chromex series. And Thora chose as her 3 PIs Abraham Krikorian, and Mary Musgrave and Norman Lewis. These are really giants in the field of plant space biology, and each is a past president of this society. So very distinguished, and there were 6 plant growth chambers within this single mid-deck locker hardware. Each PI team received 2 plant growth chambers. I worked with Abe. I was kind of his right-hand man for setting up the experiment. And I can just report to you that Abe's doing well. He's 84 years old, and he's retired and enjoying life on Long Island. And so he's doing well. Sadly, Mary Musgrave developed a brain tumor and passed away several years ago, way too young. She was fantastic. And Norman's right-hand man at the time was Gerard Hayenga, who many of you, especially from Ames and BioServe, know well. But Gerard is no longer with us. He's alive, but nobody knows where the hell he is. Norman's team also flew an experiment in the plant growth facility. This was in 1999, which was the successor to the plant growth unit. But today he's going to tell you about his experiment in the advanced plant habitat. He was the first peer-reviewed PI to fly within that facility. Norman.
Well, thank you very much, and good morning to everybody. I always like to make sure that people can hear at the back. Hear okay? Being Scottish, I always ask, or always point out, you don't need to understand, just whether you can hear is all I'm asking, yeah? So anyway, as Howard so nicely, introduced this set of experiments we're doing, involves, of course, a large team aspect here, ourselves, although on this working now, working a second ago, right? Anyway, the team going from left to right is ourselves with Laurence Davin, who's sitting in the audience here. We do the metabolomic side. Folks at Los Alamos, Sean Starkenberg, and Dick Sayre, were involved in transcriptomics. Mary Lipton, Kim Hickson, proteomics, Dave Hansen, photosynthesis, and also doing some measurements, as I'll point out. And of course, the team at KSC, Howard and Brian Oneta, and Oscar and Jeff Richards, Nicole, J.C. Carver, were involved in every aspect of these experiments that we were doing. And then we have the two astronauts, Serena and Randy Arnold, I think was his name. So let me move on to the next slide. So I like to use this slide to remind that the evolution of all forms of life, particularly when I point out with respect to plant life and lignification, plants making their way from an aquatic beginnings to terrestrial land occurred maybe about 850 million years ago. But during that time, a huge number of species have come and gone. And of course, today we listen to people talking about climate change and COPs meetings about the dangers that face us, today. But the aspect I want to focus upon is lignification, which is the sort of building block that helps plants on land be able to stand upright, to be able to produce conduits with which they can transport nutrients and water from the roots up to the uppermost portions of the plant. And the lignin also function as a protective barrier to opportunistic pathogens. And lignins are basically nature's, at least terrestrial, second most abundant biopolymer. So that's the aspect that I'm going to be focusing on today. The plant species we use is the model plant species Arabidopsis. It's a fairly short-lived species that lives for about 8 to 10 weeks. It goes through different phases of development, beginning on the left-hand side with cotyledons, then forming these rosettes at the bottom, which you can see at 4 weeks, rosette leaves, and then these uppermost aspects are the stems, which are lignifying, which we're going to be mainly focusing upon, and that then, of course, keeps continuing right through to the stages of maturation and completion. So looking at the stem aspects, the issue here is that the lignifying components are shown in here as sort of vascular bundles. Between these vascular bundles are what we call interfascicular fibers, and you can stain the lignin with chemical stains there, and you can highlight these as shown there on the right-hand side at the ages of 3 and 7 weeks. And the lignin contents in there will be about 22-23% of the plant material. We're focusing here on the gene family called the rogenate dehydratase. It's a 6-member gene family, and from that it produces phenylalanine, and that phenylalanine, prior to plants colonizing the land, would be used for making proteins. But in the 6-member gene family, 5 of these 6-member families produce a whole series of the plant phenolics, including lignin. So if you push on this button, something should be happening. It's not happening yet. So these are the four genes that we focused upon. Arabidopsis gene 2, the ADT2, is involved in protein synthesis, but these other ones are involved in making plant phenolics, including lignans. And when we start knocking these genes out, we reduce the ability to produce the monolignals and lignans and the lignin content itself, as well as some of the other pathways that are involved in this as well. So then, if you look at what we next did, we used, we got a whole number of Salk homozygous knockouts or knockouts at that point, and we generated 15 single, double, and triple knockouts, and these are distinguished from the wild type. Single numbers is a single knockout, dual numbers are double knockouts, triple knockouts, quadruple knockouts, etc., as well as overexpression lines. So there are 15 of these we generated. And you can see the phenotypes that are produced on the right-hand side. The wild type is the stems are upright and looking fine. But as you go from 1 to 2 to 3 to 4, down at the bottom, quadruple knockouts, you can see that the plants can no longer stand upright. They're sort of lying flat on the surface where they're growing. And if you look on the left-hand side, you can see that the lignin reductions that are in the plant go down by 70% from wild type down to the corresponding quadruple knockout. And the question we had was, could we reduce lignin content since in microgravity, the requirement for lignification might not be the same as it is on Earth. You would certainly anticipate that to be the case. So just to give you an idea, with some of these gene knockouts, whether we'd put them into these so-called KEGG categories. You can see examples here of transcripts, metabolites, and proteins. The coloration here, green to red, goes for whether you're seeing increases or decreases. And this is just one example of just showing that there's not always a good correlation between transcripts, proteins, and metabolites that are there. And of course, that's quite well known now. And we can segregate these things into KEGG functional categories. And you can see here in the red that we had significant effects on things like glucosinolate biosynthesis, photosynthesis, linoleic metabolism, phenylpropanoid metabolism, etc., all shown here in red. And again, you can put these functional categories into whether they're knockouts or overexpressed lines, ages that you can see that we looked at these plants from 2 weeks, 4 weeks, 6 weeks, etc. So the point I would like to make here in this particular slide, and let me tell you that these slides with these KEGG categories violate every rule of making a slide that I'm aware of. Yeah, they're unreadable, etc. Blah blah blah. But the key point to point out is that when we knock out the 4 genes, the ADT 3, 4, 5, 6, these pathways shown on the top, you can see increased attempts of the plant to increase the levels beyond the orogenate dehydratase to try and generate more of the monolignals. And of course that doesn't, and then lignin, but of course that doesn't happen. And when we do the overexpression in the green, that goes in the opposite direction. So that's the point that I'm trying to make here. So then, when we turn to the Arabidopsis experiments, we had chosen the 6 lines here, wild type, and single, double, quadruple mutants that would reduce lignin contents, as well as 2 others to try and increase biomass, which is carbon capture mechanisms, as well as a double mutant there with what we call AD3, 4, 5, 6 CCM. And you could see again, just to remind you what these phenotypes look like on the 1G on Earth, and the reduction in lignin contents that we know. So the purpose here was to have 2 grow outs, 2 harvesting times at 4 and 6 weeks, And because of the number of plant samples, et cetera, when we got the samples returned, we had to establish by PCR that the samples were in fact what they were labeled as. Yeah. So here's the timetable that we began. The SVT was in 2017, and we had a couple of EVTs as we went through trying to optimize how the APH worked. The installation of APH was in 2017. It was tested, as Howard showed you. And I want to just point out that when we did the first grow out, I actually watched that from a train from Xinjiang through the Gobi Desert, that I would FaceTime up with J.C. Carver, and he would then hook us into what was occurring on ISS. That's just an interesting aspect of how we can use communications in ways that we never thought before. Then we had 2 grow-outs in 2018. Samples were returned basically a year apart in January of 2019, 2020. And then, of course, we ran into COVID-19 with the delays that are associated with that. So the aspects here, this is just a picture of one of the EVTs. You can see the Arabidopsis plants here growing upright as best they could. And then the experimental design we had, of course, was to take these plant lines, the various ones that I've shown you there from wild type through, harvest them at the various time points, separate them into stem and leaf tissue. We unable to collect root tissue there, and then have these set up for the various multi-omics, phenomics, transcriptomics, proteomics, lipidomics, metabolomics, etc. So the interesting aspect is when we ran the Kennedy Space Center SVT and EVT experiments, we plotted the lignin levels here relative to the maturation that we have seen in lignification, in our experiments here. And you can see that in the SVT, which is in the open squares and circles relative to the EVTs. At first, the SVT plants weren't growing as well as they might. There was some tweaking there to get the EVTs to function much better. But the stage that we were focusing on was during this active lignification phase, which is before the system plateaus. So everything looked quite nice at that point. So I'm going to ask Jim just to click this video on again. And you've already seen this a few seconds ago. But the point I'm trying to make is the unusual curvature of some of these stems, and I'll come back to that in a few minutes. And these experiments were all just done with wild type systems. So once we turn now to the actual experiments on the plant habitat, you can see that the pictures on the right here show different growth times from 1 week through to 6. And Dave Hansen's group measured the area of these rosettes, that the sort of leafy tissue at the base of the plant. And you can see, quite interestingly, that in the 2 grow-outs, the Kennedy Space Center ground controls grew much faster than the corresponding plants on ISS. And that didn't matter which system, whether it was grow-out 1 or grow-out 2. And we were trying to get the plants to grow to the same level of maturation. The interesting aspect here was that with these plants in the first grow-out and the second grow-out, the plant systems that had the ADT knocked out still would not stand upright, and during the growth of the stems eventually touched the bottom of the APH carrier, indicating that even with the 70% reduction and not even 70% reduction of lignification, the plant stems themselves weren't able to stand upright in the same way as the corresponding wild type and CCM experiments did. The important aspect to note here is that when we looked at the Arabidopsis cross-sectional differences between the ground controls at KSC and ADT 3456, and I'm sorry about this pointer not working, but on the very right-hand side there, we can see an aberration in the vascular apparatus there that does not exist in the ground controls and does not exist in the experiments with both the wild type and the ADT 3456 at Kennedy Space Center. So the whole vasculature has been reprogrammed in some way on that particular, on these flights. So then when we looked at the KSS and ISS APH Arabidopsis lignin levels in the first grow out and the second grow out, the first grow out, the plants grew a little bit better, and you can see the lignin contents here. The ISS is in the open circles and squares, etc., relative to the KSC ground controls. And in each case, the KSC plants outgrew or outperformed the corresponding ISS experiments. There was significant variability on growing the plants in the different sites within the APH growth chamber, and that gave rise to a little bit of what you might call biological variability. Okay, so then we went to the multi-omics, and these were all the different omic forms that I've just described. The first aspect we would do would be metabolomic analysis, where we looked at many plant metabolites. This just shows one chromatogram. The areas that we were focusing on were the post-phenylalanine phenolic metabolites, phenylpropanoids, flavonoids, phenolic glucosides, but there were also other systems like defense molecules like glucosinolates and galactolipids, apocarotenoids, etc., that were all affected. And we did this in the leaf and the stem, KSC versus ISS. And using partial least squares discrimination analysis, you can very readily segregate out I'm just showing the examples here of the wild type plants and the ADT 3456. You can segregate these out. They are shown in the right very readily to establish which phenotype is which and which one experienced spaceflight and which one was the ground control. In the proteomics side of things, this was all done at PNNL. You can see there's about 16,499 unique peptide-derived proteins. And again, you can see this segregation in using PLSDA analysis into whether it's on leaf or stem or whether it's in ground control or on ISS. And in a similar way, differential gene expression gave the same. There were 1,403 differentially expressed genes. Okay, so we then wanted to focus on the steps leading up to the phenylalanine. And again, this shows you the changes which occur. Red indicates an increase, black is more or less no increase, and green is a decrease between KSC and ISS. And all I can tell you is that the plants, whether they were in KSC or ISS, displayed fairly similar characteristics. These are not huge differences between the two plants grown in space and on space flight. Similarly, when we look at the phenylpropanoid pathway, and the phenylpropanoid pathway is making the lignin biopolymers as well as a whole bunch of plant defense molecules, you can see again KSC in the red here, which is decreased relative in many of the pathway steps. Some are in green that show some upregulation and the corresponding in ISS. But the important thing to note is there's really not a huge difference between the two sets of experiments. And when we look at the quadruple knockout versus the wild type KSC, we can see that on the left-hand side in the red there, the ADT genes are sort of that are picked up out of the fragments. You don't get the ADT actually formed, but you can pick up some of the ADT aragonate dehydrogenase gene family member fragments there. You can see these are down, and in the blue, the post-phenylalanine, the phenylpropanoid pathway enzymes are all increased, but not, again, not resulting in being able to restore these levels. It's an abortive attempt to try and increase the amount of lignin and other moieties that are being generated. And when we look at the ISS versus KSC, it's interesting there that we see a different effect on the lower end. This is, we see a decrease in these particular genes in the blue shown there as the during the ISS coverage. So the plants in this case are not trying to do the same as they were doing on land, but again, these are not hugely different systems. Then when we look at integrating the transcriptomics, the proteomics, and the metabolomics, we're just going to give you this an example here in leaf and stem tissues, where we look at the proteomics in the P, transcriptomics in the T, for all the different lines that are there, and then the effects they have on this group of metabolites that are shown there. Again, there are differences are noted. There's some asterisks there and some lines showing a more significant effect. For example, in ADT 3456 at the top there in the leaves, and then ADT 5 on the stems. But the overall effects are still quite comparable between the ground controls and the ISS. And we can do the same here for the flavonoid pathway. We can look at these systems here. And this is really one of the big challenges in terms of trying to understand multi-omics experiments. It's not just getting the data from the transcriptomics and the proteomics and the metabolomics. I grew up with doing biosynthesis, so to me, the metabolomics is the final answer of what that organism will do, regardless of what type of organism is, and then we have to go back and try and correlate that with the transcript and the proteome data. There's clear effects that you can see on ISS, I guess you would call radiation damage, repair mechanisms that come into play more higher than they do on Kennedy Space Center ground controls, and you can see the types of things that are involved here, from mismatch repair, etc. I won't read all these things. You can see them down the side or download these at a different point. And similarly, cell signaling related proteins, we can see significant differences in the induction of these on ISS relative to ground controls, and similarly transport-related proteins. So that's all at the protein level. Then, if we turn our attention, though, to genes differentially regulated in leaf tissue during Wright, without going through the different series that are here, you can break them down into defense, plant defense, radiation, stress response, water stress, etc., and cell wall structure. And similarly, in the mutants, genes that are involved differentially regulated in the mutants, comparing all the genotypes and using, of course, the go biological processes, you can see radiation, stress response, and water stress. So in summary, and then I've got a few other slides I'll just quickly go through. In summary, those plant systems, even they grow a little bit slower on space station, really grew nicely. They came back looking healthy, and they generally grew well. They were a little bit slower on ISS than they were at KSC. We could see that we were able to reduce the lignin contents quite significantly, but these plants were still unable to get their stems moving up into an upright position, the ones that were downregulated in lignification. And you could see from what I've just skimmed through here, there's a negative correlation between the lignin reduction and proteins involved in DNA repair, cell signaling, transport proteins, etc. So you may also say, now when we knock out these genes so that phenylalanine cannot be processed into either structural biopolymers or plant defense molecules, have we made the plants more susceptible to microbial invasion, for example. And that's a question that one needs to ask about. But the other aspect also might be that when we downregulate ADT, perhaps we're also triggering these defense systems, but they might not be of any real significance. And the question that I have about long-term space duration travel and colonization on Mars or the Moon, etc., is how you're going to be able to regenerate plants, recycle that carbon, nitrogen, oxygen, etc., and be able to do it. And to me, one easier way of doing this would be to have plants with lower lignin contents that you can then utilize and regenerate, the elements for regenerating plants in the systems. So I'll stop at this point. I will also say that, we've done the same, compared KSC, ISS for plant hormone signal transduction, for photosynthesis, antenna proteins. You can see, look at the differences and similarities. For carbon fixation, differences, similarities from ground to control. Glycosylic and dicarboxylic metabolism. The ribosome has differences that we noted already, and we see them again at KSC, ground control, and ISS, and also in the spliceosome. So the point I'm trying to make is that it's been a great honor to be able to have been the first folks to have experiments on APH. We've got a massive amount of data. We're writing up these papers as we speak just now, and still continuing to process and interpret the data. So at that, thank you very much for your attention, and sorry the pointer wasn't working. Yeah.
All right, we have a couple minutes. Quite a few questions actually came in for you, Dr. Lewis. So I think in the interest of time, I'll just grab a couple of them. But one of the questions that came in early on in the talk was, do you know why your antenna proteins are so heavily affected in your mutants?
Say that again, please.
Do you know why your antenna proteins are so heavily affected in the mutants?
I think we can say that it's a phenomenological difference that we note, and that we are viewing the results that we have as a starting point to go in to investigate and understand how these processes occur. But I will say that one interpretation that we have is that photosynthesis itself is not necessarily shut down. So CO2 conversion into sugars and then into other metabolites, we wondered that when we did those experiments, would that CO2, if it was still being taken up, would we generate more plant material, more carbon being put into plants, but not being put into lignin? Well, that did not occur. And what we believe is occurring is that while the photosynthetic apparatus is being upregulated, photorespiration is removing that carbon so that you end up not having the same levels of carbon that are in there from wild type to the mutants. Does that sort of answer your question?
And maybe one more quickly. Since the lignin plants didn't stand up in space, is there an impact on light tropism?
On light what, sorry?
Light tropism?
Tropism, is that what you said?
Yes.
I'm not in a good position to answer that question. I think what I can say is that the plants themselves are unable to stand upright. The mutants that we have from ADT5 through to the quadruple mutant at 1G. And we've seen that, whether it's greenhouse grown or growth chamber or APH grown. It was our working hypothesis at the time that with the stems growing in space and microgravity, that lower lignin content would not affect them being able to stand upright. That turned out not to be the case. And as we think about it, we also wonder, because remember that these lignifying cell types in the vascular bundles have got to be able to transport, water and nutrients through the reinforced lignified cell types. And if that failed in any way, then plants wouldn't be able to take up water or transport nutrients there. So whether you're looking at some mechanism there whereby plants still, lie sort of prostrate on the surface or make their way towards the surface of the APH. What we don't know very well is what the effects of that lignification has on the cell wall structural properties of these. And of course, that could also be affected by light tropism as well, but that's not something we've given much thought to. We're still trying to understand why the lower lignin content plants just don't stand upright in general. But you can see that, like any good research, it's asked more questions than you began with, and there's room here for an awful lot of further investigation. Does that make any sense?
Thank you. Thank you, Dr. Lewis. We just don't have time to answer anymore, but I did get, Dr. Lewis, quite a few questions for you, so I'll pass these along. Let's thank Dr. Lewis one more time for an excellent talk.